Hey guys, in this video we're going to talk about the second domain of life, which is bacteria. Again, these are prokaryotic cells and they have peptidoglycan, unlike archaea, found within their cell wall. We can use the composition of peptidoglycan to characterize two main groups of bacteria, gram-positive and gram-negative bacteria, and we're going to look further into that in later slides. And a key characteristic of bacteria is the fact that they do not have histone proteins for their DNA, unlike both archaea and eukarya. And they have a 70S ribosomal composition because they're a prokaryotic cell, but it's different from archaea, and because it's different, we can target that using antibiotics like streptomycin. Lastly, one thing that's unique for bacteria are endospores. You can kind of think of an endospore like a seed. So it's hard, tough, and it's just dormant in the external environment until conditions are right. So these can be dormant for hundreds, even thousands of years. And during this time, they can withstand a wide variety of adverse environmental conditions. However, when conditions are right, you could think of it like hatching. They'll open up to germinate to form a bacterium cell. Bacteria is classified based on its shape. So this is found in the nomenclature, what we use to refer to a specific species of bacteria. And there's three main shapes found in bacteria. Number one is bacillus or bacilli, that's the plural form, and this is rod-shaped bacteria. So it looks like a small rod-shaped pill. The next is coccus, and the plural is cocci, or if it's two, diplococcus or triplococcus. So whenever you have the word coccus in there, these are spherical shaped bacteria, so they look like a ball. The last type of shape is spirillum or spirilia. These are spiral-shaped bacteria, so they look like this off on the right-hand side here. Once we have identified the shape of the bacteria, we have to see how it groups up. When you have bacteria found in a chain-like fashion, we use the word strepto as a prefix. So you'll hear the word streptobacillus or streptococcus. Strepto meaning chain, bacillus meaning rod-shaped, or streptococcus, meaning spherical-shaped bacteria that's found in a chain. So this over here on the right-hand side is streptococcus. Another type of grouping is a cluster of bacteria, and that is known as staph. So when you have staphylococcus, that's a cluster of spherical-shaped bacteria. So right here, this is staphylococcus. Now we get to a microbiological topic that's commonly used by taxonomists to classify two types of bacteria. The first one is gram-positive, and the second one is gram-negative. So you have gram-positive bacteria and gram-negative bacteria. So what makes bacteria gram-positive or gram-negative? Well, for gram-positive bacteria, it means it has a thick layer of peptidoglycan. Remember, that's a key molecule used for classifying bacteria. Now this cellular wall of peptidoglycan, super thick for gram-positive bacteria. However, superficial to that, sometimes you'll have an outer membrane called a lipopolysaccharide outer membrane. For gram-positive bacteria, that is not present. Then the last characteristic that you should know is between the thick layer of peptidoglycan and the actual cell plasma membrane, gram-positive bacteria are going to have teichoic acids present. So these key characteristics of gram-positive bacteria cause it to turn dark purple when it is stained or gram stained. So microbiologists use a gram stain, a test to see if bacteria is either gram positive or gram negative. And if that bacteria turns purple, it means it's gram positive, which means it has a thick layer of peptidoglycan, it does not have an LPS outer membrane, and it does have teichoic acids present. So think P, purple, positive, that's gram positive bacteria. Gram-negative bacteria, on the other hand, have a very thin layer of peptidoglycan found in their cell wall. However, they do have a lipopolysaccharide outer membrane superior to that present. That means they also have endotoxins present. That's a type of lipopolysaccharide. These are released when the cell is killed or when it disintegrates. Then lastly, there is no teichoic acids present between that peptidoglycan layer and that plasma membrane. So these characteristics right here cause gram-negative bacteria, when it's gram-stained, to turn pink. So if you have pink bacteria shown, you know, okay, it's gram-negative, it has a thin layer of peptidoglycan, but it does have that outer lipopolysaccharide membrane present. And if it has a LPS, a lipopolysaccharide outer membrane, that means there's no teichoic acids. So here's just a visual representation of gram-positive versus gram-negative bacteria. Off here on the left, we have a thick layer of peptidoglycan, and then superficial to that, there is no outer membrane, no LPS present. So this is gram-positive bacteria off on the left-hand side. Off on the right-hand side, we have a thin layer of peptidoglycan here in purple, and we have this superficial membrane, this LPS present. So this is gram-negative bacteria. Here are two common types of bacteria that sometimes show up on the DAT. Number one is nitrogen-fixing and nitrifying bacteria. Nitrogen-fixing bacteria and nitrifying bacteria, these are key players in the nitrogen cycle. So nitrogen-fixing bacteria takes atmospheric nitrogen, so N2, in our atmosphere. They take that in and they fix it to ammonium ions. Nitrifying bacteria will use those ammonium ions and convert them to nitrate. And nitrate can be assimilated by plants, so it's taken in by plants. So that's why they have this mutual realistic relationship with plants in the nitrogen cycle. Another common type of bacteria that's tested on the DAT is cyanobacteria. Now this is a classic trick question because cyanobacteria is also referred to as blue-green algae. And we're going to learn that algae is eukaryotic, so eukaryotic cell type. But cyanobacteria is not eukaryotic algae. It's just another name for photosynthetic prokaryotic bacteria. So when you see the terms blue-green algae, just automatically think in your head, okay, this is actually bacteria that they're talking about. This is cyanobacteria. This is not eukaryotic algae. One topic that you should be well versed in with the domain bacteria is the types of genetic recombination. So there's three ways, and we're going to look at that right now. So number one is conjugation, two is transformation, and the third is transduction. Okay, we're going to quickly gloss over this. So the first is conjugation. This is sometimes referred to as horizontal gene transfer. Again, this is unique to prokaryotic cells. The thing that you should know about conjugation is it involves a transfer of a plasmid. Plasmids are separate from the host genome, so it's not found in the nucleoid, and it is accessory DNA. Plasmids can be transferred via a pilus. So this is what a pilus looks like. If you have a plasmid, you're going to have the genes to code for a pilus. So this bacterium off on the right-hand side does not have a plasmid, so it does not code for a pilus. Why do bacteria cells want to transfer plasmids? Well, it's because a plasmid can house resistance, unique genes that will make them more fit in their environment, and overall increase the survivability of a bacterium cell. So that's why you would want to transfer a plasmid. So what's going to happen is the pilus is going to be inserted into the other bacterium cell that does not have that plasmid. And then this plasmid is going to be copied over into the new bacterium cell. So because this new bacterium cell has that plasmid, it's going to have the gene to code for a pilus. And then this process can continue on. So that's conjugation. A bacterium cell can also uptake plasmids found in the external environment. This is huge. This is a huge breakthrough that we have discovered in producing certain genes, like insulin, for example. To put it very simply, transformation is going to involve either heat or calcium chloride, CaCl2. That's going to cause the bacteria cell wall to open up a little bit, have some pores to uptake those plasmids in the environment. So when you heat shock a bacterium cell or you add calcium chloride and you have plasmids found in the external environment, that bacterium cell is going to take in those plasmids, and that is known as transformation. The last type of genetic recombination that you should know for bacteria is transduction. This is going to involve a virus. Now we're going to look at what a virus is in the next slide, but what you should know here is that a virus can inject its own genome into a bacteria cell. And this bacteria cell can incorporate that within its own genome. So this is a way to get foreign DNA into a bacterium. Once that genetic information is incorporated into the genome or the plasmid, it can be transferred to other bacteria cells through conjugation, for example, using a pilus, or passed on through different generations through binary fission.